The method of choice of nucleic acid quantification in all areas of molecular biology is real-time qPCR. The method is so called because the implication of DNA with the polymerase chain reaction is monitored in real time. There are two methods that we can use to detect qPCR products in real time. Method 1 uses non-specific fluorescent dyes that intercalate with any double-stranded DNA. This is what we use in our experiment.
Beginning with qPCR, we start with the dissection of tissue. This year we focused on gills because it's the first line of defense for mucosal exposure. We also decided to use viscera because it tells us more about the systemic immune response. After the tissue is dissected, we have to be sure to put it in RNA later to preserve RNA. Then we isolate the RNA. Once the RNA is isolated, we have to be sure to make it into cDNA rather quickly because the RNA can break down. Once we have our cDNA samples, we will dilute those samples and then place them in a PCR plate. Then the PCR plate can go through the PCR machine.
So after obtaining the mean CQ values from the qPCR, we calculated the DCQ and DDCQ. To do this, we standardized all the samples by subtracting RPL13, which was our housekeeping gene, as well as the control. We compiled these DDCQ values into histogram and line graphs and graphed the relative expression of IgZ1, IgZ2, IgM, and BAFF. From this data, we observed that the immunoglobulins and BAFF were most highly concentrated within the viscera of the fish, and we realized that little expression was